All right, everyone, welcome. Today we're diving into something that's absolutely central to biochemistry, biological oxidation. We're going to explore the amazing story of how our cells generate the energy that powers, well, everything.
Okay, so check this out. The source material for this actually personifies the star of our show, ATP. It's like ATP is introducing itself. I am the energy currency of the cell. I mean, that sounds pretty important, right? It's definitely the central player in our story today.
So that's the big question, isn't it? What exactly is this energy currency and how in the world does the cell go about making it? To get there, we've got to start with the basic rules of the game. So let's kick things off with bioenergetics. And look, don't let the name intimidate you. It's basically just the study of how energy works in living systems. Think of it as the physics behind all the chemistry we're about to talk about.
All right, so first concept, free energy, or delta G. The simplest way to think about this is it's the amount of energy that's actually available to get work done. It's the key number that tells us if a reaction is going to happen by itself or if it needs a little push. And here's the bottom line, the thing you absolutely have to remember. If delta G is negative, the reaction releases energy. We call that exergonic. It's spontaneous, it just goes. But if delta G is positive, it needs energy. It's endergonic. This one simple rule, guys, it runs all of metabolism. It's that important.
Okay, so we've got the rules of the road. Now let's meet the superstar molecule that pays for pretty much everything that happens in a cell, ATP.
So ATP belongs to a class of molecules we call high-energy compounds. These are basically little chemical batteries. They're special because when you break them down, they release a significant chunk of energy. While you've got others like phosphoenol pyruvate or creatine phosphate, ATP is the universal one, the main player.
And this right here shows you where the magic is. See those two little squiggly lines? Those represent high-energy bonds. Honestly, the best way to picture them is like tightly coiled springs. When the cell needs to do something, say make a muscle contract, it just snips one of those bonds. The spring uncoils, and boom, energy is released to do the work.
And this brings us to the ATP-ADP cycle, which is happening constantly. Think of it like this. Your cell spends ATP, breaking it down to ADP to get work done. Then it uses the energy from the food you eat to recharge that ADP, sticking a phosphate back on to make ATP again. It's this nonstop cycle of spending and earning, happening trillions of times every single second in your body, right now.
All right, so how exactly do we recharge that ADP back into ATP? Well, the main factory for this is inside our cellular power plants, the mitochondria. And the assembly line inside is called the electron transport chain. Let's go see how it works.
So, let's zoom way in. Inside our cells, we have these amazing little organelles called mitochondria. You've probably been calling them the powerhouses of the cell since middle school, right? Well, today we're going to see exactly why they deserve that name.
So what's really going on inside this powerhouse? Well, on that super folded inner membrane, you've got this incredible assembly line called the electron transport chain, or ETC. This is where most of our ATP gets made. But, you've got to get the fuel to the factory first, right? The fuel is a molecule called NADH, but it can't cross that inner membrane on its own, so it has to use these special shuttle systems, like the malate aspartate shuttle, to get its electrons inside. So picture this. NADH and FADH2, which we get from breaking down food, they're like little delivery trucks full of high-energy electrons. They pull up to the ETC and drop off their cargo. These electrons then get passed down the line, from one protein complex to the next, kind of like a hot potato. And with each handoff, a little bit of energy is released. And at the very, very end of the line, oxygen is waiting to pick them up. This, right here, is the whole reason you're breathing.
Okay, so these are the names you're going to want to know. These are the key workers on this assembly line. You've got these big stationary complexes, 1, 2, 3, and 4. And then you have these little mobile carriers like Coenzyme Q and Cytochromes C that act like shuttles moving the electrons between the big guys. It's just an incredibly organized, perfectly synchronized relay race.
All right, so we've got this flow of electrons, and it's releasing energy. But how does that energy actually get used to make ATP? Well, that is the process of oxidative phosphorylation. It's the grand finale, where the oxidation part, the electron transport, is finally coupled to the phosphorylation part, making ATP.
And the explanation for how this works is called the chemiosmotic hypothesis. Peter Mitchell came up with this, and it's such a beautiful and elegant concept it won him the Nobel Prize. Let's break it down. It's really just 2 steps.
Okay, part 1. Build the pressure. As those electrons are flowing down the chain, the energy they release is used to actively pump protons from the inside of the mitochondrion, the matrix, into that little space between the 2 membranes. What you're doing is creating a massive proton gradient. It's exactly like pumping water uphill to fill a reservoir behind a dam. You're storing up a huge amount of potential energy.
And now for part 2, generate the power. We open the floodgates. The protons want to rush back down into the matrix, and the only way they can do that is by flowing through this incredible molecular machine called ATP synthase. And as the protons flow through it, they literally make a part of it spin, just like water spinning a turbine. That spinning mechanical motion provides the energy to physically jam a phosphate back onto ADP, recharging it into ATP. It is just a mind-blowing piece of natural engineering.
Okay, now let's just pause for a second and think about this. This whole system is so finely tuned and so essential for life. So, what happens if it gets blocked? Or if something goes wrong? This is where this biochemistry becomes incredibly relevant for you as a future doctor.
Yeah, so certain poisons work by basically sabotaging this assembly line at very specific points. You've got rotenone, which blocks complex I. Antimycin A hits complex 3. And the really famous ones, cyanide and carbon monoxide, are so deadly because they shut down complex IV, the very last step. When that happens, the whole chain backs up. Electron flow stops, proton pumping stops, and ATP production just grinds to a complete halt.
Now, uncouplers do something different, but just as dangerous. They don't block the chain. Instead, they poke holes in that inner mitochondrial membrane. So remember our dam? It's like they've just drilled holes in it. The protons can now just leak back into the matrix without going through the ATP synthase turbine. The proton gradient is gone, the electron transport chain actually goes into overdrive trying to compensate, but all that energy is just lost as pure heat. This is exactly why that banned diet drug DNP caused a deadly fever.
So you can see how this stuff is just critical, from understanding poisons and toxicology to genetic diseases. For example, your mitochondria have their own little circle of DNA. And mutations in that DNA can lead to some serious inherited disorders. A classic example is Leber's hereditary optic neuropathy, which causes blindness. Why? Because the cells in your optic nerve have massive energy needs, so they're one of the first things to fail when your ATP production is faulty.
Okay, quick pop quiz. Let's see if you were paying attention. A patient comes into the ER and you suspect cyanide poisoning. Which specific protein complex in the ETC is being blocked? Think back to that table. You got it? It's cytochrome oxidase, complex 4. See? Knowing these details connects what you learn in the classroom to literally saving someone's life.
And there you have it. You guys did a great job. You've just walked through the absolute core concepts of how our bodies create and use energy. This is a huge foundation. Keep coming back to these ideas because you're going to see them pop up everywhere. In physiology, pharmacology, pathology, you name it. You are well on your way.